Hello, grade 8 students. In this video, we'll talk about the revision for the semester 1 exams. This semester will include the immune system. So let's start with part 1, immunology, chapter 1, the immune response. First activity about the self and is divided into is defined as the army that protects our body against the harm around us, like the bacteria, the viruses, the parasites. So the immune system, as I said, is the army. It contains soldiers. We're going to determine these soldiers later on. So the immune system is the army that protects our body against the harm, against the bacteria, against the viruses, against the parasites. So without the immune system, we will die. So immune system is like an army in the country. For example, in Lebanon, we have the Lebanese army. It's very important. So without the Lebanese army, we're going to have a lot of problems. So here, the immune system is like the army inside our body. So it has the role of protection. So the immune system, al-mana'a in Arabic. So this is important for the protection against the bacteria, against the viruses, against the parasites. So the immune system, let's start with the role of the immune system. What is the function of the immune system? What does it do? So the role of the immune system is to defend the body against any possible invasion by the bacteria, viruses, and parasites. So coronavirus is called COVID-19. So here you have to be careful about this. We're going to have a question similar to this, COVID-19. So the role of the immune system is to defend the body against any possible invasion by bacteria, by viruses, and parasites. So here, immune system defends, attacks citizens, foreigners. So as a conclusion, the immune system will defend or it will attack the citizens or the foreigners. So to start with, it defends that belong to my body. So my immune system defend my organs, my cells, my tissues. So the immune system will defend. We have two verbs, defend, attack. Okay? Defend or attack. So the immune system will defend the citizens, meaning that belong to my body, or the foreigners that are different from my body. Sure, the immune system will defend my citizens, my organs, my tissues. So defend is going to be for the citizens. So the immune system will defend the citizens and it will attack the foreigners. So the foreigners will be attacked by the immune system. The immune system will attack any non-self. So as a conclusion here, we have to say that the immune system defends its own citizens while it attacks any foreigners. So here is the conclusion, the first conclusion, and the very important conclusion here, that the immune system defends its own citizens, meaning that the immune system defends its organs, defends my tissue, my organs, my cells, while it will attack any foreigners. In the example, it will attack Ali's lung, because Ali's lung is a foreigner with respect to Ahmed's body. So it's classified as a foreigner. So here, citizens, we're going to call them as self. Self, they belong to my body, to myself. While foreigners, they are classified as non-self. They don't belong to myself. They don't belong to my body. HLA. HLA, the definition, it's called the human leukocyte antigen. H for human, L for leukocyte, and A for antigen. So human, leukocyte, antigen. Where it's found? It's found on the surface of nucleated cells. What's the meaning of nucleated cells? Nucleated cells mean that they have nucleus. So what is HLA? HLA is the human leukocyte antigen. It's found on the surface of nucleated cells. It's found on the membrane of nucleated cells. What is the role of HLA? Why do we have to have HLA? HLA is the biological identity marker that helps the immune system to distinguish self from non-self. Let's say, for example, we have football club. Let's say we have Real Madrid and Barcelona. Okay, so Real Madrid and Barcelona, both of them, they have different logos. This is the logo for Real Madrid and this is the logo for Barcelona. So these two logos, they are different. How can we determine that this player belongs to Real Madrid on the field? By having the logo of Real Madrid and having the shirt of Real Madrid. So having the logo of Real Madrid, you can know that this player belongs to Real Madrid. Having the logo of Barcelona, you know that this player belongs to Barcelona. So HLA, it's like the logo. For example, having the logo on your shirt for ISI, you know that the student belongs to the International School of Innovation. So by this way, we can determine the student belongs to the ISI. Here, biological identity marker, this will help the immune system to distinguish the self from non-self. If this cell have the HLA, so it belongs to my body. So it's a self. So the immune system won't attack it. It will be safe. So the immune system depends on the HLA. So HLA is the biological identity marker that helps the immune system to distinguish the self from non-self. So if we have the HLA on the cell, meaning that it has the logo, it have the marker, it have the identity marker, okay? So this cell belongs to my body. Another example, you have the Lebanese ID, okay? So whenever you pass, you have this ID, meaning that you are Lebanese, so you can pass. If you don't have your ID, meaning that you are not Lebanese, you can't pass. So this one is called an example about the biological identity marker. So the biological identity marker, it helps the immune system to distinguish the self or non-self. If you have the HLA, meaning that we have the self cells. If they don't have the HLA, they are non-self. So, third note, the cells of the same body and those of identical twins, they have the same HLA. So for example, my heart, my kidney, my liver, my stomach, for example, my eyes, my skin, and others, they belong to my body. So they are the cells of the same body. So they have same HLA. Also, identical twins, meaning that identical twins, they look like each other. Okay, so identical twins, they have also the same HLA, while the cells of different persons, for example, the cells of Hussein and Ali, they don't belong to each other. They are, for example, friends only in the class, so they have different HLA. So as a revision, what is HLA? It's a human leukocyte antigen. It's found on the surface of the nucleated cells. It's the biological identity marker. It's the marker, and it's specific for the biological identity marker that helps the immune system to distinguish the self from non-self. So if it's self, it should have the same HLA. If it's not self, it should have different HLA. Move now to some notes about the HLA. Each person has a specific HLA, on the surface of nucleated cells, the cells that have nucleus. But wait a moment, we have special types of cells. They don't have nucleus inside our body. What do we call them? They are the red blood cells. The RBCs, red blood cells. They don't have nucleus, so they don't have HLA. That's why we can donate blood for any person. So we don't have HLA. So the immune system will not attack these red blood cells and will not consider them as non-self. Why? Because they are anucleated or anucleated, meaning that they don't have nucleus. So red blood cells, pay attention to this note, it's very important. Red blood cells, they don't have a nucleus, so they don't have HLA, because we said that HLA is found on the surface of nucleated cells. Also, identical twins, as I said before, they have identical HLA on their nucleated cells. So now let's develop Freire's model for self and non-self using the HLA. Now this is the complex definition for the self. Here, Freire's model, here we have the term in the middle, here we have the definition, here an example, here a non-example about them. Okay, so let's start first with the term self. What the meaning of self? We said before that self is all that belongs to one's body, like my hand, my kidney, my stomach, and others. Here we added a few terms or few words and has the same HLA. So whenever you ask about the term of the self, you have to say that all that belongs to one's body and they have the same HLA. Without same HLA, meaning that you don't know the definition of self. So self, all that belongs to one's body and they have the same HLA. Self, example, my hand, my kidney, my liver, my pancreas, my eye, my nose, and others. They are called self. Here another example, my friend's hand. My friend's hand is not self. It belongs to others' body. It's called non-self. So as revision, what is the self? Self is all that belongs to one's body and has the same HLA. Example, my hand, my liver, my pancreas, my kidney, my stomach, my eye. Non-example, my friend's hand, my friend's kidney, my friend's nose. All of them, they are non-examples. Okay, let's move now to the definition of the non-self. So the non-self. What is the non-self? Definition. The non-self, all that does not belong to one's body. And here we have additional information here, and they have different HLA. So what the difference between self and non-self? We said that the self is all that belongs to one's body and they have the same HLA. while the non-self, oh, that does not belong to one's body, and they has different HLA. Example about the non-self: my friend's hand, my friend's kidney, my friend's lung. Non-example, my hand. This is my hand. Example about the self. So here we have the definition of the self and non-self. You have to know them. Self, oh, that belongs to one's body and they have the same HLA. Non-self, oh, that does not belong to one's body and they have different HLA. Now let's move to an idea about grafting. What is a graft? A graft is a transplant of a tissue or an organ from one side to another in the same body, or from one body to another. It's moving from an organ. You know that some people, they can donate a kidney, for example, or they can donate a lung, or they can donate, for example, let's say an eye. So this is called grafting. So grafting is the transplant of a tissue or an organ from one side to another. For example, some people, they have, for example, problems in their face. They have, for example, burned their face, for example, by fire or something. So they have damaged skin cells. So they can take skin cells from the thigh, from the leg, and they can transplant it into their face to have the recovery for the skin. So this is from one side to another in the same body. Or, for example, we can transplant an organ from one body to another. For example, I can donate a kidney for my sister, or I can donate a liver, for example, or I can donate, let's say, a lung to my brother, let's say. So this is from one body to another. Here we have different types of grafts. Let's start with the first type. First type is called autograft. Auto definition is the graft that is transferred from one side to another in the same body. So it's transferred, the graft is transferred, or the transplant is transferred from one side, from one position to another position in the same body. Example about this: from my hand to my thigh, either from my hand to my leg, or from my thigh to my face, let's say. This is an example. Non-example: from my hand to my friend's hand. This is a non-example. Okay? Another type of graft is called isograft. Isograft is done between a person and the identical twin. So always I, identical, I, iso. So I, identical twin, isograft. I, identical twin, isograft. Here, Samir's kidney transferred to his identical twin. Maybe it's called Fadi, for example. So isograft is the graft that is done between two identical twin. This is an example. Not an example: Samir's kidney transferred to his sister twin. Sister twin, pay attention. Sister twin is not an identical twin. So pay attention to this. They should be, both of them should be males, or both of them should be females, in order to say that they are identical. So identical twin, the graft that's done between two identical twins, is called isograft. So till now we have two different types of graft. The first graft is called autograft. The graft is transferred from one side of the body to another side in the same body. And we have the second type, isograft, the graft that is transferred from one person to the identical twin. That's why I, identical, I, iso. So I, identical, I, iso. So you can easily detect this. Let's move now to the third type of graft, called allograft. This graft, allograft, is done between one person and another in the same species, and they belong to the same species. For example, from Hassan to Maya, from Ali to Rewa, from Celine to Sabine, from Sabine to Zahra. So all of them, they belong to the same species. Okay? Here, for example, the graft is transferred from one person to another in the same species. Okay? So this is called allograft. Here, example about this: from Maya to Rola, from Hassan to Hassan, from Karim to Hadi, from Hadi to Rida, from Rida to Abbas, from Celine to, let's say, to Fatima. All of them, they belong to the same species. While an example, from a cat to a dog, cat and dog, they belong to different species. So it's not an example about allograft. The last type of graft is called xenograft. Xenograft is a graft that's done between two different individuals that belong to different species. For example, from a cat to a dog, from a dog to a horse, from a horse to, for example, let's say zebra, from zebra to lion. Okay, so they belong to different species. From, for example, from a human to a dog, let's say, or from a dog to a human, they belong to different species. So this is an example. From a cat to a dog, cat and dog, they belong to different species. Cat and, for example, a hen, let's say. So they are different species. So here we have 4 types of graft. Autograft, within the same body. Auto, within the same body. Iso, iso, identical. Allo, different individuals in the same species, from a human to a human, from a cat to a cat, from a dog to a dog, from, for example, a horse to another horse. They belong to the same species. While xenograft, they belong to different species, from a cat to a dog, from a dog to a hen, let's say, from a dog to a human. So they belong to different species. So what affects the fate of the graft? What affects the result of the graft? Fate is the final result. What will happen to the graft? What affects the result? So the fate of the graft, whether it's success or failure, it depends on the similarity of HLA. This is the most important note. So the graft is accepted or rejected depending on the similarity, of the HLA. If they have similar HLA, so the graft is accepted. If they have different HLA, so the graft is rejected. So the graft rejection or acceptance depends on the similarity of HLA. As the similarity of HLA increases, the percentage of success will increase. This is done between a donor and the recipient. What the meaning of a donor? Is the one that donates. So let's say, for example, I want to donate a kidney to Mr. Ali. So who the person will donate? I am the one who's going to donate. So I am the donor. Mr. Ali will receive this kidney. So we call Mr. Ali the recipient. So the donor, the one that donates. Here's the animal. Mostly is done on animals. So you can say the one that donates is called the donor. Meaning donate, it means gives. And the one that receives is called the recipient, the one that takes. So here, recipient and donor. So the fate of the graft, I repeat it, depends on the similarity of HLA between the donor and the recipient. Okay? Hope this video will help you to understand every single note about what is immunology, what is immunity, what is the role of the immune system, what is HLA, what are the different types of grafts. So watch this video and hope it will help you, and thank you for watching this, and see you soon. Bye bye. Let's move now to the blood groups, or what's known by the ABO system. According to the blood typing system, which is the ABO system, we have four different kinds of blood types. We have blood group A, blood group B, blood group AB, and blood group O. So let's start first with the blood group A. Always the blood group will have the same antigen as the blood group. For example, blood group A will have the antigen A, while the antibody will be the opposite of the antigen. For example, let's start first with the blood group A. Blood group A, if you belong to blood group A, you have antigen A on the surface of your blood cells as shown here. But you have antibodies B in your blood plasma. While blood group B, if you belong to blood group B, you have antigen B, while you have on your plasma, you have antibody A. Blood group AB, so you have both antigens A and B on the surface. You have antigens A and B on the surface of your red blood cells, while you have no antibodies in your blood plasma, since you have both antigens. So no antibodies should face or should encounter them unless we will have agglutination. Blood group O is known to be the null group. You have neither antigen A or B. So there is no antigen A or antigen B, but we have antibodies A and antibodies B in the blood plasma. Group O is known to be the universal donor, while blood group AB is known to be the universal recipient. This table is very important to know the blood transfusions, who can receive blood from whom. So blood group AB, which antigens? So they have antigens A and antigen B. Antibodies, there is no antibodies. So who can give? Blood group AB can give only to AB, but they can receive from AB, from A, from B, and from O. So they can receive from all blood groups. That's why it's called the universal recipient. So they can receive blood from the all blood groups. Blood group A, it has antigens as the blood group, so it has antigen A, while antibodies they are B. So blood group A can give to A and AB, and they can receive blood from A and O. Blood group B, they can have antigens B and antibody A. They can give blood to B and AB. As you see here, AB, they get blood from all the different blood groups, while the blood group B, it can receive from B and O, while O, there is no antigens. They have antibodies A and antibodies B, and they can give blood to AB, and O. So AB can get blood from the all blood groups. That's why it's called the universal recipient. While blood group O, it can donate to the all blood groups AB, and O. That's why it's called the universal donor. While the antibody, the blood group O, can receive blood only from O. Let's move now to the immune cells. In the cells, we have different blood cells, different components, and different functions. Let's talk first about the red blood cells. Red blood cells, they are known as the erythrocytes. Erythrocytes, they are small in size, they are many in number. They are red in color, and they are anucleated, meaning that there is no nucleus, and the role is to transport oxygen and carbon dioxide. Whereas the white blood cells or the leukocytes, they are large in size and they are few in number. They are not red in color, they're nucleated. Their role is to defend the body, and they are the immune cells. So let's talk about the cells involved in immunity. Wait a minute, the cell, it looks like a horseshoe shaped. It has a horseshoe shaped nucleus. So what do we call the cell? The cell is called the monocyte. Monocytes, they are the cells involved in immunity. They have horseshoe shaped nucleus, they have a cytoplasmic membrane, and a small cytoplasm. So large in size. This is the most important characteristic. They have a horseshoe shaped nucleus. They flow in the blood vessels. They can cross the capillary wall, and they have a role in phagocytosis after maturing into a macrophage. So they have a role in phagocytosis, eating other cells. These cells, they are called granulocytes. Why? Because they have granules in their cytoplasm. They have a nucleus which is a multi-lobed nucleus. So the characteristics, they are large in size, they have a multi-lobed nucleus, they have granules in their cytoplasm, they flow in the blood vessels, and they can cross the capillary walls, and they have a role in phagocytosis. Moving now to the lymphocytes. We have two different types of lymphocytes. We have B lymphocytes and we have T lymphocytes. Both of them, they have large round nucleus and they have a small cytoplasm. What differentiates the B lymphocytes? They have antibodies. So their role is to secrete antibodies to kill the non-self. B lymphocytes labeling, you have large round nucleus, the small cytoplasm as we said. These are the antibodies, the Y proteins, Y-shaped proteins, and we have the cytoplasmic membrane. T lymphocytes, they have the same properties. Small cytoplasm, large round nucleus. They have a large in size and they are called the killer cells. Why? Because they kill other cells using this structure, which is called T cell receptor. T, C, R, to kill the non-self. Moving to the immune organs, we have the primary immune organ and the secondary immune organs. They are the organs of the production and maturation of the immune cells, the primary, while the secondary, the sites of the immune response. Immune organs, they are divided primary immune organs and secondary. Primary, they are the bone marrow and the thymus. Secondary, they are the lymph nodes and the spleen. Bone marrow produces all the immune cells, produces B and T lymphocytes, and matures only B, while the thymus have an only role in the maturation of T. So bone marrow is the source of the B and T lymphocytes. If there is no bone marrow, there is no maturation of T lymphocytes in the thymus. So bone marrow is very important for production of B and T, but it only matures B. Lymph nodes, they are the site where the immune response against tissue infection occurs, while the spleen is the site where the immune response against blood infection takes place. Moving now to the natural barriers. What are the natural barriers and what is the function of each of these? Let's move now to talk about the natural barriers. They form the first line of defense. So what would happen if these natural barriers were broken? If the first line is broken, what will happen? We will have infection. It's the invasion and the multiplication of the pathogens in the body. Second, we have the pathogen. What is the pathogen? It's any non-self that infects the body, like bacteria, viruses, and nowadays we have the virus and is capable of causing a disease, is classified as a pathogen. Inflammatory response is the immune response against tissue infection, against any infection in the body. It is the second line of defense. This is what we call by inflammation. Inflammatory response, we have four signs. We have redness, we have heat, we have swelling, or it's called edema, and finally we have pain. Signs of inflammation, we have to know that the first cells to cross the capillary walls are the monocytes and the granulocytes that have a role in phagocytosis. That's why they are called the phagocytes. Characteristics of the inflammation, it's instantaneous and it's non-specific. What the meaning of instantaneous? It starts immediately. What the meaning of non-specific? It doesn't depend on the identity of the intruder. Steps, the phagocyte approaches the pathogen. Here are the steps of the phagocytosis, eating other cell. The phagocyte approaches toward the pathogen. Second, the phagocyte will adhere to the pathogen and gets in direct contact with it. These are the steps of phagocytosis. You have to study them. And number three, the phagocyte will engulf, ingest, and digest the pathogen. So main actor of the phagocytosis, they are the phagocytes, they are the monocyte, and the granulocytes. They are the phagocytes or the macrophages. Phagocytosis is known to be the non-specific immediate immune response against infection. Its actors, they are the phagocytes. Now let's move for specific immune response. All what we talk about inflammation, about the phagocytosis, we are talking about the non-specific. Moving to the specific immune response, we have first the first line of defense, they are the natural barriers. The second line of defense, they are the non-specific immune response. And the third line of defense, what do we talk about this? It's the specific immune response. So what are the steps of the activation of T lymphocytes? Steps of activation: first, the specific T lymphocyte will approach toward the target and the infected cell. Then the T lymphocyte, what will do? The T lymphocyte will adhere, will bind to the infected cell using the TCR, and then it will give this cell the killer kiss. This will perforate it and will create a hole in it. As such, the cell dies. What is the type of the immune response carried out by T lymphocyte? It's called the specific immune response. While what is the name of the immune response carried out by T lymphocyte? It's called the cell mediated immune response. T lymphocyte cell mediated immune response. So therefore, the T lymphocytes, they carry out specific cell-mediated immune response, where the T lymphocytes, they kill other cells in both cases. Type of the immune response carried out by B lymphocyte, also its specific immune response. Why the name? It's a humoral immune response. So the B lymphocytes, they carry out specific humoral immune response, where the B lymphocytes, in this case, they secrete antibodies. So whenever we have secretion of antibodies, meaning that the immune response is humoral immune response. What are the characteristics of the specific immune response? Primary immune response and the secondary, what's the difference? You have to keep in mind that primary immune response is the immune response upon the first contact with the pathogen, whereas the secondary immune response is the immune response upon the second contact what with the same pathogen. So you have to keep in mind here that multiplication, it means proliferation, it means amplification. The antibody producing cells, they are called plasma cells. During differentiation, a cell differentiate either into effector cells and memory cells. Effector B cells, they are called plasma cells. They are able to secrete antibodies, while effector T cells, they are called the killer cells. They kill other cells using their TCR. This graph is showing the rate of the antibodies in the blood in respiratory unit as a function of time in days after the first injection of the antigen, which is called the primary, and the second injection of the antigen, which is called the secondary immune response. As you see here, the immune response has a long latency. Latency is the time needed for the immune response to start. It needs about 10 days to start secretion antibodies, while in the secondary, it has short latency. It starts after 2 days. Primary immune response, it has low amount of antibody. It's about 2 AU, while the secondary it's about 6 AU. High amount of antibodies during the secondary. Also, short duration of protection in the primary, while we have long duration of protection in the secondary. So here, a table showing the comparison between primary and secondary immune response, it shows that the latency, the time needed for the immune response to start, it's long latency, long period, while short in the secondary, low amount of antibodies in the primary, while high amount in the secondary. Duration of the response, it's short, while in the secondary, it's a long duration of protection. So you have to keep in mind that the primary immune response takes a long time to eliminate the infection, yet it produces the memory B and T cells. These memory B and T cells, they are going to be used in the secondary. That's why it will be more efficient and it takes less time due to the presence of the memory cells from the primary immune response. So sure, primary immune response, it takes a long time to eliminate the pathogen, but it produces the memory B and T cells needed in the secondary to be more efficient and to be faster and takes a shorter time.